and also the kind of initiate the R&D in order to produce a prototype and to prove that it's stable and you can work with it really, to help with supplying the basic information required to respond to IP issue and regulatory issue, to keep good records, that's very critical in a proper way, to supply industry with all the needs for advancing the program smoothly, and to interact and to be open with the industry. And this is our case, it's me, the TTO is Isum, this is the story of Doxil, and let's see how I evaluate it. Okay, TTO Isum of the Hebrew University at that time. Well, what kind of help I got really? Financial and package, the TTO gave me nothing. I have to find everything myself. They didn't find me anything. Help on IP issue, they did. That's very good, I think, did a good work on it. Help in finding the funding, they didn't. Again, I did all by myself. Help in contractual arrangement with respect to IP issue and all this aspect, yes, they did partial. I also got some other advice, luckily. Guidance on how to keep records, no, they didn't know how to do it at that time. So actually we were teaching them, and what we taught them now is used by all the applied scientists in the Hebrew University. Help in presentation, no, I have to do by myself. Criticism, well, they accepted all what I said, which not always the case. So this was a long time ago. Now this company is much better. They learn a lot from my experience. But I dealt with many TTO that I have, because we have dual agreement, and I have to say that at the time that it was done at the 20th century, all the TTO of most universities were not better than this one. Now we start going to Doxil. What's all about? What's Doxil about? It's about this actually balance. We can see the balance between toxicity and efficacy. We're trying to improve efficacy and reduce toxicity. That's actually the story of Doxil. Doxil is highly toxic. Every organ you touch in the body, Doxil will damage it. You know, alopecia in the hair, mucositis, cardiotoxicity is a very major issue, myelosuppression, anything. You can see a slide I took from Alberto. You can see here actually anything. And the idea is, how can we kind of reduce this toxicity and improving the efficacy? And that's taking us back to the very famous scientist, the Paul Ehrlich, with the magic bullet idea, how we can change from the distribution of the drug all over the body, including our target disease tissue, to a distribution that most of the drug will be accumulated in the disease tissue and very little will be in other organ. By this we improve efficacy and reduce toxicity. And there are a few ways to do it. And I'm going to talk in one way, and this is the way that taking advantage of the disease itself, or the anatomy of the disease, and the metabolism, as I show you later, how to take advantage of it and make sure that most of a lot of the drug is localized really at the site of the tumor, the disease site, and also that something in the tumor site acting as a signal that enables the drug to be much more effective. We'll talk about it. But there are other ways to do it. We are not going to talk about them, including just local injection at the site like you do when you're dealing with local anesthetic. But we'll focus today on this situation. So there are many drug delivery systems we have. Nano-drugs, we focus on nano-drugs because nano-drugs are the only one useful for systemic use. That you injected IV, and that's why I focus on them. And in tumor, that's what we're dealing with mainly, is with the systemic disease. So there are many kinds of the question, well, why to select liposomes? So the reason now, because liposomes are biocompatible, biodegradable, they are not immunogenic. We know a lot about them, the scientific community. They've known pharmacokinetics and biodistribution, and the lipids of the liposome have a known metabolism. So from point of view of toxicology, they have the advantage over most other nanoparticles where things are not that known about them. That doesn't mean that the other nanoparticles will not find their way to the clinics and to the market, and maybe they will be even better. But this required time, and this required all you young guys really to do the work and try to get to the same level of knowledge we do have with the liposomes, and that takes a long time. The fact that I said that liposomes are known doesn't mean we don't anticipate toxicology. Actually, it's a slide I made in 1993, I think, in a review that we wrote together with Herod Storm, and you can see here that actually many toxicity are expected in nanoliposome, and actually they show up. And we know today about complement activation, and we know about something related to the MPS, and some of them we know can be some immunogenicity, some related to quality of dispersion. But as Patrick said, I think we also know that we can overcome all of them. So you really have to know how to do it, but once you know, I don't think it's a major obstacle. But we have to be aware that such a toxicology really can exist. Liposome. Well, liposomes were discovered, they are not invented, they were with us since the beginning of life. They claim that one of the claims is life started in liposome, actually, the evolution in life, but this guy is actually Alex Bangham, Professor Alex Bangham, when he was a pathologist in the British Army in Israel, Palestine at that time. You can see him here. And that's what he gave me as a present when I was a drawing of him that I put here that show how we make liposome, and you can see how the phases, when you take a lipid and phospholipid, hydrate in water, you can see the phases it will go, how it will till he make a liposome. And what he did, he actually encapsulate potassium, radioactive potassium, in this multilamellar liposome, and then he put in the dialysis bag. He removed all the external potassium and actually have a liposome loaded with potassium that can be used for whatever you want. And this was actually a sign that you can really have a drug delivery system. And although Bangham never dealt with drug delivery system, he wrote it already in his first review in 1965 that maybe because they imitate cell membrane, they may be actually not recognized by the immune system, and therefore they can serve as a good drug delivery system. So that's not much change from there with the ability of Alex Bangham to encapsulated molecule, except that we do it much better. Now I'm going to share with you a short movie about Doxil development and then we go back and end the story. So let's see. It has sound here? Can you have sound? I'll start it again. Okay, so actually the movie it's a 6.5-minute movie, and it goes from the idea of Bangham till the mechanism of action of Doxil. And we made it actually in order to explain Doxil in a popular way. I think it's really summarized the story very well.
The human body is made up of billions of cells. Each cell is wrapped in a membrane, whose function is to selectively screen what goes in and out of the cell. The basic building block of this biological membrane is phospholipids, fatty molecules made up of a polar head, which has an affinity for water, and a fatty hydrophobic tail, which is insoluble in water. The head is usually composed of glycerol and phosphate. The tail is composed of two long chains of fatty acids. Molecules which have both hydrophobic and polar groups are referred to as amphiphiles. Another essential fatty component is cholesterol. The membrane also includes proteins, which are responsible for many of the membrane's unique functions. Due to their amphiphysy, phospholipids tend to stay in the interface between the two environments, oily and watery. The basis of all biological membranes is the formation of the lipid bilayers, which are organized so their heads face the watery phase and their tails face each other. In the 1960s, Alan Bingham of Cambridge University discovered that membranes can be created artificially. He took phospholipids, dispersed them in a watery solution, and discovered that these molecules can self-assemble spontaneously, forming a bilayered vesicle which has one or more phospholipid bilayers, enclosing trapped water inside it. This structure is called a liposome. Into the internal nanoenvironment, different molecules, including drugs, can be encapsulated. There are several kinds of liposomes. Today, liposomes can be custom-made. In a laboratory setting, it is possible to predetermine the liposome size, electrical charge, shape, and number of lipid bilayers, single, few, or many. It is also possible to track fat-soluble materials in the liposome's membrane and water-soluble materials in the water trapped inside the liposome. Liposomes have several uses. The study of membranes for one. But the most important practical application is in the nanopharmaceutical industry as nano-drugs, a new category of liposome-based pharmaceuticals that are particularly effective delivery systems for drugs. When we take a drug orally or intravenously, it reaches our bloodstream.
counter ion with methanesulfonate, and actually what this doing, we can see in the X-ray we don't have crystals. In the calorimeter, it's a very different type of structure we have. Release rate we get is slightly faster, and this actually caused a major improvement in the performance, as we see here. This is a PPE of Doxil, and this is equivalent dose PPE of our new formulation in Rats model, and you can see it's much better. So we now finished the toxicity, and we plan to take it all to clinical trial and really see if it really worked better. And needless to say that we checked that it didn't lose efficacy and did not affect the other aspects and gains we get about toxicity of the Doxil. So I think at the end, I would like to just to show the teams that involved with Doxil. You can see many groups and the companies, the clinical people in Israel, the clinical people in other places. Franco Muja, I want to really give him a lot of credit. He was the MD that really handled the clinical trial in the US. Very important, the most experienced physician with Doxil in the world, I guess, today. Papadopoulos, a friend of mine, died unfortunately, but really is the one that really encouraged me to go on to the development of Doxil. You can see my team, Alberto team, and Sequest. Every one of them is a head of team, so it's many people involved. And these are people that did the new work about Doxil. Lisa, that developed the ammonia, Shawe, the DSC, and other things, Dima, that did part of the QC, and other things, these things I didn't show you, and my collaborators, Uri Raviv, with whom we developed the XRD work. And I want to end my lecture with a motto. You know, here we are at the area of Confucius, I guess, isn't it? That's what I was told. And I loved Confucius when I was a child. I always admired the things he said. I think it's a lot of wisdom of life. And one of the things I like very much is choose a job you love and you will never have to work a day in your life. And that's actually how I feel. Thank you very much.
Thank you very much. If you have time schedule, you can keep it short. Okay.
Very interesting talk. I didn't hear anything about the Doxil directly changing basic mechanism of Adriamycin that is inhibiting topoisomerase, so that DNA cannot make a macromolecular compound. That's the basic mechanism. You seem to explain based on ETR effect, but was there any study related to this kind of enzyme effect? Is compared to just a free Adriamycin, was there any more inhibition of topoisomerase?
Okay. Well, I think the answer is very simple. Actually, what we do with the Doxil is we bring more doxorubicin to the site of the tumor and we release it there. Once we release it, doxorubicin acts like normal doxorubicin. There is no difference. It inhibits topoisomerase, it do everything the same. Because we delivered more, we can actually do a better job. Not as good as we want, and I think I stated it very clearly. If you look, this is the data from a phase 2 clinical trial comparing topotecan against Doxil. Okay? Now, the reason topotecan was there as a comparator is not because we want it. We didn't want it. We want to do it against Doxil, but FDA said topotecan is better than doxorubicin. So you have to compare it to topotecan, not to doxorubicin. And that's what we did. And actually, you can see that in this model, it's not true for all the tumors. But in the ovarian cancer, we are better, Doxil is better than topotecan. So, but the mechanism of doxorubicin stayed the mechanism of doxorubicin. We changed the pharmacokinetics and the biodistribution, and by this we reduced cardiotoxicity, and you can see it here very clearly, and I show it in the slide with one patient, but this slide done on many patients, and you can see this is the green is the toxicity, cardiotoxicity, very low cardiotoxicity of Doxil, and actually we can go up to 3 times this and still the cardiotoxicity remain at this level, while with doxorubicin here, you already have a prohibiting toxicity. So we reduce the toxicity dramatically, and as we know, doxorubicin is a very good drug, but it has a limit to its capacity for 2 reasons. Resistance, which we did not overcome with Doxil, and cardiotoxicity, which we did overcome with Doxil. So, as I said, and I said it in the beginning, if you ask me if I'm happy about Doxil, I said yes, but I'm not enough happy. I want to have it to do much better, and I believe now, and I'll show it in my lecture on 2 days or 3 days from today, 2 days from today, that we can do better now with animals, and now we are moving into the clinical trial. But basically, the mechanism of action of doxorubicin itself is the same. Okay?
Okay, thank you very much. Thank you. We will have a short break for 10 minutes until lunch. Thank you.
Thank you for your help. You were responding very fast and nice.
요거 연결하면 네, 이거는 아까 이게 뒤에 자료 여기
네.
있는데 위에 있습니다.